Morning. I want to make an announcement about the midterm, so those of you can get on your schedules. There's going to be a review session for the class for the exam, which will be held in this room on the Wednesday which precedes the midterm, and it's going to be from 6 to 7 in the evening. So if you want to come to the review session, it will not be webcast. It will not be simulcast to the two side rooms. This will be the only room in which you'll be able to listen to the review session. And I'll talk more about the review session as we get closer to the time. But I wanted to mention it to you now so you can get it on your calendars so that you are able to come if you wish and you have the notice. Last time we ended by talking to you about secondary growth and about this very interesting but really poorly characterized layer, which we said was the vascular cambium. And we said that as a result of the cambial division activities that we produced secondary phloem to the outside and secondary xylem to the inside. Secondary xylem, as you know, consists of dead cells when functional, whereas secondary phloem, when we talked about the phloem, consists of living cells but one type without the nucleus. So the question I want to pose at the very beginning is, here we have a single, presumably, cell layer, which to its outside makes one cell type, and to its inside makes another cell type. Both of these cell types are very different. Both of these cell types come from a common initial. And what we want to begin to study today, and we'll continue discussing on Friday, is the commitment to particular developmental pathways. In other words, we want to ask, what is it that sets the procambial cell or the vascular cambium cell on this side off to differentiating as a secondary phloem versus what are the circumstances or the developmental events which cause the cells to differentiate as secondary xylem, which is ultimately dead. Now, the studies of this commitment process, of this development process in plants, goes back to the mid-19th century to a supposedly very dogmatic German plant physiologist by the name of Sachs. Now, Sachs believed that plants had specific substances which were targeted to doing certain developmental events. So he believed in the notion of tissue or organ forming substances. And his views were that there was a root forming substance, a shoot-forming substance, etc. That there were lots of different substances which accounted for the various organ and cell types in the plant. Now, at the time that Sachs was putting forward his ideas in Germany in the 1850s, 1860s, two English workers, one of whom you know fairly well, Charles and his son Francis, were also interested in trying to understand commitment and development in plants. And for their work, they started to use a tissue system that those of you who have had the lab this week have already been exposed to, and those who will have the lab today and tomorrow will see what the tissue system was that Charles and Francis Darwin used. They used grass seedlings. And in particular, we'll draw the grass seedling for you. The grass seedling, again, is a monocot, just to remind you. And in particular, what they were interested in studying in the grass seedling is something that many of you have seen already. They were interested in the sheath, or the coleoptile, in this particular grass seedling. So they were interested in trying to understand developmental aspects of the coleoptile. And let's talk about what their initial interest was. Both of these individuals, who eventually published a book, and I'll give you the name of the book in a moment, on this subject, both of these individuals were interested in plant movement, or as it is known scientifically as tropisms. And the tropism that they were particularly interested in studying with the coleoptile, and we'll talk about their experiments in a moment, was phototropism, which you've already been exposed to when we talked about Pilobolus, the fungus. That is movement in response to light. And in particular, what they were interested is if they took a coleoptile seedling, and these experiments were done in darkness, or in very dim light, but we'll talk about more of the conditions in a moment. So if we do these experiments in darkness, what they found is that the coleoptile coleoptiles grew straight. Now, if they took the coleoptile seedling and they gave it light from one direction, which is known as unilateral illumination, if they gave this light coming from this direction, they discovered that the coleoptile grew toward the light. That is, it bent toward the light. This is phototropism. Now, what I'd like to do is just to magnify this response on this coleoptile bending toward the light to make an important point. In order for this coleoptile to bend or to grow toward the light, the growth that we would see on these two sides would differ. We would have more growth on the dark side, and it's really not dark, on the dark side, and less growth on the lighted side. This difference in growth, which was important for the response, is known as differential growth. And we're going to be seeing several examples of differential growth as we go through the discussion today. Now, Charles and his son did a lot of experiments on coleoptiles, trying to understand the developmental basis for phototropism. And perhaps the major result to come out of their work was from experiments in which they took coleoptiles, and again they did initially grow them in the darkness, and what they did is they asked what region of the coleoptile perceives the light. And to do these experiments, and I'll show you some pictures, they covered various regions of the coleoptile with some kind of a dark material, and then they exposed the coleoptile to light, and they discovered that it was the tip which perceives the light. And they theorized, although they had no evidence for this, they hypothesized that some signal must come from the tip. So these experiments were done in the late part of the 19th century, and Darwin and his son Francis published a book with these and many other observations. The book is called The Power of Movement in Plants. Of which we have a number of copies in our library. It is full of very interesting observations, many of which have not been followed up. These observations on phototropism were made, as I said, in the late 1800s, and they were then just left to lie and not be further explored until about the 1930s, when people took up these observations using what were said to be more modern techniques to try and understand more about the phototropism process. And the kind of experiment which was done at that time involved first trying to establish or to reprove what Charles and Francis had concluded, namely that the tip was important and something was coming from the tip. And so these individuals in the 1930s, again, used grass seedlings, just as Charles and Francis did, and used coleoptiles, but instead, what they did is they put a barrier between the tip of the coleoptile and the region which was responding. And when they put this barrier here and they gave unilateral illumination, that is light from one direction, they were able to confirm basically what Francis and Charles had concluded, that there was no bending. So the conclusion was that some substance coming from the tip acted at some distance from the tip, that is below it. In another region of the coleoptile. And that this substance was present in very small amounts. All you needed was very small amounts. These observations that the substance was needed in very small amounts and that it was acting at a site which was different from its apparent site of formation led to the conclusion that this substance, in the animal sense, was a hormone, because it was formed presumably in small amounts and acted someplace differently from its site of formation. Plant physiologists prefer not to use the word hormone because it implies a lot about the mechanisms of action, and instead we use the word plant growth, I should write this out, plant growth regulator to describe material that people have initially called a hormone. As a result of being able to confirm Charles and Francis's observations, these individuals in the 30s went on to do a large number of experiments, and I want to just give you a few of the key experiments they did and to give you some conclusions. The first experiment of the many, although this may not have been the actual first, is they did this in darkness. And I should say that it isn't really darkness. They do these experiments in very dim green light, because green light doesn't appear to be involved in the phototropism response. So it allows us to see what we're doing, but it doesn't allow the plant to see any light. So in darkness, what they did is they took coleoptiles, and they, in one sense, they took the coleoptile and they removed the tip. And then they took another one, also in darkness, and they left it intact. And then they waited for some time. And in this case, where the tip was removed, what they found is that there was essentially no elongation. And in the one that had the tip intact, there was a dramatic increase in elongation in the coleoptile. So again, suggesting that even in darkness, something important was coming from the tip. Based on this understanding, or at least hypothesis, that something was coming from the tip, which was important in the growth of the coleoptile, a number of further experiments were done, and these led to the following type of experiments. On the assumption that something was being produced in the tip, which both Darwin and these workers in the 30s concluded, they wanted to ask, would it be possible to manipulate this substance? And so what was done was to take sheets of agar, and onto these sheets of agar, they put coleoptile tips. So these are tips, and they're on agar. And the thought was that if there was something coming from the tip, it would move into the agar. And we could call this something, or maybe it's many things. We could just call it a diffusate. So after allowing the tips to sit on the agar and presumably allowing whatever material was being made in the tip to move into the agar, they then took the agar and they divided the agar into squares. And then did the following sequence of experiments. And again, these experiments were done in green light or with use of lateral illumination after the coleoptiles were growing. In the first experiment which they did, they took the coleoptile and they cut off its tip, just like we did before, and they put one of the agar blocks on symmetrically. Put a block on symmetrically. They had a control, obviously, and the control had an agar block lacking diffusate. Lacking diffusate. And they waited for some time. And what they were able to show is that there was growth in the coleoptile in the dark. This is in dark. Growth of the coleoptile, which had the block that had the diffusate in it. You got growth. But no growth here. Then they did a very clever series of experiments. In this experiment, what was done was to take our grass seedling, just as we did before, to remove its tip, which is the source of this diffusate. And again, these experiments are done in the dark. And they put the block on now asymmetrically. Asymmetrically. And they waited for some time. And what they were able to show is that the coleoptile bent. This is in darkness without any light. The coleoptile bent. It showed differential growth. In the final experiment, I wanted to just bring to your attention, and as I said, there were many, many experiments they did, was the following. They took the seedling now, and they did this initially in darkness, so we're essentially repeating the experiment we have on the far left, and they put the block on symmetrically. And now, this was the really interesting clue that they did. They gave it now unilateral light. They gave it unilateral light. That is light from one direction. And when this was done, what they found was that the coleoptile, even though it had the block on symmetrically, bent toward the light. So from these experiments and many others, some of which are discussed in your textbook, there are a couple of general conclusions that we can come to. One we've already mentioned, that is that they reproved or they confirmed the observation of Charles and Francis, namely that a substance or substances, they didn't know at this point, is made in the tip Secondly, that the substance moves down from the tip, and it moves down uniformly. And I want to explain to you what that means. Let's look at an intact coleoptile growing in darkness. That is no unilateral illumination. So if we have a seed with an intact coleoptile, what that second conclusion says is that the substance or substances which are being made in the tip move down uniformly. So this is uniform movement. And since this substance seemed to be associated with the lengthening of the coleoptile, we can assume that the concentration of the substance was equal throughout the coleoptile. The third conclusion they made, which is probably the most important and the most insightful from this work, is that unilateral light causes a higher concentration of substance or substances, again they didn't know what it was, on the dark side. And this stimulates differential growth. Stimulates differential growth. So in this particular circumstance, compared to the dark one, if we have the coleoptile and we give it unilateral light, what they're saying is that there was some mechanism which they didn't understand, and in fact wasn't proven until several decades later, which caused more of the substance to be on the dark side of the coleoptile, and that this was the reason why we had this differential growth. Now, we had this substance, or was substances, diffused into the agar block, and as you might expect, the agar block was then subjected to chemical analysis. And coming from this work was the discovery of the first plant growth regulator, which was able to be extracted from the agar block. And this plant growth regulator, which was extracted and actually caused the differential growth, was shown to be a low molecular weight compound, less than 200, known as indole 3-acetic acid, or as it is abbreviated, IAA, or as it is talked about in your textbook and other places, the common name is auxin. Now, of all the growth regulators we have, auxin profoundly affects hundreds, if not thousands, of different types of plant developmental events. We don't understand how it works. I'm going to give you a suggestion a little later on. It is a substance which is able to do many things at the same time. How do we separate these two or three events to decide which one goes and which one doesn't? Because auxin does so many things, what I want to do is just give you the tip of the iceberg, a couple of examples of some of the major phenomena that some of you have seen or will be familiar with regard to auxin physiology. And the first physiological event which is regulated by auxin is apical dominance. This is a result of IAA being made in the shoot apical meristem. And here's an example of apical dominance. In apical dominance, the shoot apex, which is at the tip of this branch, is making auxin. And what apical dominance means is that the axillary buds are inhibited from growing out. So if you could remove this apex or damage it, like I'm doing here, and then wait for some period of time, as happened naturally to this branch over here, what you would find, the shoot apex was at the end of this dead branch here. For some reason it was damaged. What you would find is now the inhibited axillary buds have grown out. They are no longer being supplied with auxin from the shoot apex, and they grow out. Very complicated developmental event which is linked to auxin. Apical dominance, and we'll talk about apical dominance a little bit more. The second aspect of development, which we've already touched upon, is that activity of the vascular cambium is greatly affected by auxin, and we're going to talk about this a bit more later on. The third aspect, which is a general one that is affected by auxin, is that auxin causes branch roots to initiate. So if you have a cutting, for those of you who are gardeners or who have parents who are gardeners, and you have a cutting and you want to get roots on this cutting, you can buy a synthetic auxin in the nursery. And I have one of these synthetic auxins here. It's called Root Tone. And if you dip the wet end of the cutting into the auxin, what you are doing is you are stimulating the formation of lateral roots. Very important, but again, a very complex phenomenon that we don't understand. Auxin is involved, and we'll talk more about this toward the end of the class, in fruit development. And in particular, in the form of a fruit, or the shape. Another aspect among the just top few that I want to give you, and there are many others cited in your book, is that auxin regulates the dropping of leaves from trees in the fall. Leaf drop, which plant physiologists describe as abscission. Now, how does this happen? The leaves on the tree turn color when the seasons become less favorable for photosynthesis, usually when it gets colder. But sometimes the summer is prolonged and it's not cold, and yet we're getting close to December when it's going to suddenly get very cold. How is it that the tree is going to know to program its leaves to begin to abscise, to fall from the tree, if the temperature has not told it to do so? So auxin is produced in the leaves, and it is produced in response to the length of the day. So that even though the temperature may not change in November, the day length will have changed. And this change in day length is what signals to the leaf to stop making auxin, and this cessation or reduction in auxin synthesis causes the leaves to abscise or to fall from the tree. And finally, the last of what I told you are many different phenomena controlled by Auxin is the orientation of the plant. And in this case, both the shoot and the root. Now here we have a plant stem, and the plant would normally be growing in this sort of orientation with the roots down at the bottom. If we take this plant and turn it on its side, your experience will tell you that the shoot end eventually will grow up and the root end will eventually grow down. Both of these orientation phenomena are directly controlled by auxin. We've already discussed how the root, at least, perceives the gravity. We've talked about the amyloplasts. But after the amyloplasts fall to the lower surface of the root, and they're also found in the stems, after they fall, then a series of signaling events are triggered, and they involve, in part, auxin. So let's look at this stem now, which has been turned on its side, and talk about what is going on in the stem. And I think for this one, I'll put it here. So now we're going to take this stem, which was of a vertical orientation, and we've turned it sideways. I'm just going to make the stem large for our purposes of discussion. Here we'll have the shoot apical meristem at one end of the stem, and at the other end of the stem, we'll have the root apical meristem. The shoot has been laid on its side. Now, what happens after the starch grains perceive the change in orientation of the shoot relative to gravity? Well, what we know happens is that auxin moves to the new lower surface. Moves to the new lower surface. It does that in the shoot, and it does that in the root. It moves to the new lower surface. So that ultimately, and we'll discuss this in a moment, ultimately the shoot grows up, and ultimately the root grows down. So the original orientation is reestablished. Now I want to call your attention to an important aspect of this drawing. In the case of the shoot growing up and in the root growing down, we have to have differential growth. This side is growing at a different rate from this side. But in the shoot, the lowermost surface grows more. And in the root, the uppermost surface grows more. How is this possible? What is going on to give these two different results, which presumably reflect auxin concentrations? I posed a question for you earlier when I said that auxin does thousands of things in the plant. How is the plant able to decide which of those things it wants to do and which of them it doesn't want to do? And the answer to that question is shown in the following graph, or at least part of the answer. And I'll put the screen down while we're talking. On this graph, on the y-axis, I'm going to put elongation. Here will be no change. This part will be an increase, and this part will be a decrease. And on the x-axis, I'm going to put auxin concentration. Here it's high, and here it is low. Now let's look at the root. What is going on with regard to auxin concentration in the root when the plant is turned on its side? Initially, the root is growing at some rate before we turn the plant on the side. Then we turn the plant on the side, and what we notice is that auxin is beginning to accumulate on the lower surface of the root. So there's an accumulation, and this accumulation temporarily results in an increase in the elongation of the root. But as more and more auxin moves to the lower surface of the root, the elongation becomes inhibited. So here is the root, and with auxin concentrations increasing at the lower surface, we inhibit the growth because there's too much auxin there, too much of a good thing. Let's look at the shoot now. In the shoot, we have a steady state, and now the auxin is beginning to accumulate on the lower surface of the root, and just as on the lower surface of the shoot, and just as before, as the auxin accumulates, we stimulate growth until we get too much growth, or too much auxin, and we begin to get inhibition. So what we can say for our question that we raised before is that one of the ways auxin can do many different things in tissues is by changing its concentration and by having different sensitivities to the auxin molecule. For example, at this concentration here, let's just choose an arbitrary concentration. At this concentration here, the root lower surface is inhibited from growing, whereas if we go up, the shoot is stimulated as far as its growth. So differing in concentration and sensitivities is one way we control the different auxin effects. Now, this graph has been used by many people, especially people who are developing compounds that can be used and based on auxin physiology. And a group of those compounds have been made into herbicides. They are based on the fact that we have differing sensitivities of plants to auxin. So, for example, eudicots, we could substitute the word eudicot for root here, and let's say you have a lawn with dandelions in it. And for the shoot, which we said was here, we could substitute the word grass. In this particular case, if an herbicide is developed, what you want to do is find a concentration of the herbicide, and these herbicides are based on synthetic auxin molecules, which affect development, in this case cause death, in the eudicot, the weed you want to get rid of from your lawn, and not affect the grass. This differing sensitivity is the basis for many different types of effects that are done by auxin. Let's look now at some of the auxin slides, and if you could just dim the lights, that would be great. Here is a picture of Darwin's bedroom. I've been told by people who have actually visited his home in England that this room remains the same as it was in the late 19th century. Here is the book he and his son Francis published, and as I said, we have a number of copies. This was published in 1896 here in our library. It's a fascinating book. These are the kind of experiments Darwin did. He used candles. He didn't use electric lights. And in these experiments, he blanked various portions of the coleoptile and then was able to show that the region which perceived the light was the tip. Here are the coleoptiles sitting on the agar. The diffusate, whatever it was supposedly going in. And these are the kinds of experiments which we talked about that people did. And again, many of them are discussed in your textbook. Here is an actual experiment. These are done in green light in very humid rooms, very uncomfortable experiments to do, in which the agar block is put on asymmetrically and then bending, and they usually have armies of coleoptiles in the responses to get the proper data. Here's an example of strong apical dominance in a palm tree where none of the axillary buds grow out and there's only one axis to the plant, as opposed to this plant from Africa where the main shoot apex has had very low or very poor apical dominance, and you have many, many axillary buds released from inhibition from apical dominance and growing out. Here's an example of the use of the material that I have up here called rootone in getting plants to root, and the auxin stimulates the formation of the branch roots. And here we have the example of reorienting the plant, and these two parts over here exhibit differential growth. Both have high levels of auxin on the lower surface, but in one case, the level is high enough to stimulate growth, and in another case, that same level is high enough to inhibit growth. Here's an example of a corn seedling where the high level of auxin here would inhibit growth on the lower surface, and the seed would then reorient after it perceives gravity. This picture was taken right outside VLSB before they put the central stairway in, and what I want to call your attention to is this tree has mostly lost its leaves except for the leaves that are near the lamp. And that's because these leaves are not perceiving a change in the season since they are being given a prolonged light period, and so they think it's still summer. So these are the auxin slides. It is a very interesting group of hormones, growth regulators, poorly understood. A lot of contemporary work is being done on auxins, but we really are very far from understanding how they work and how they do so many different things at the same time in the plant. Now what I want to do is to talk to you about the second group of plant growth regulators which were discovered. And this group of plant growth regulators was discovered in the 1930s by the Japanese. But because of difficulties in Americans not understanding Japanese, and then because of World War II, these results were not really known in the United States until the late 40s or early 50s. Now, what resulted in the discovery of this next group of growth regulators came about from Japanese scientists who were interested in studying a disease in rice plants. And in this particular disease in the rice plant, what would happen is the rice would get infected by a fungus, and then the rice would become very, very tall and skinny. And when any kind of breeze or heavy wind came along, the rice was blown over into the water, and the heads which contained the seeds, that is the harvest, were lost. And so the Japanese workers were trying to understand what it was about this infection that caused the plants to become long and skinny. The fungus which caused this particular elongation response and the thinning of the stems was known as Gibberella. And what the Japanese workers showed was that the fungus Gibberella was able to do this massive change or induce this massive change in development through the production of a compound which they call gibberellin. Now, this compound was first identified and isolated from a fungus, but we now know it is also naturally occurring in all healthy plants. And there are now probably in excess of 100 individual chemical compounds which are gibberellins, and they are also called gibberellic acid and are abbreviated in your text as GA. And often there is a number associated with it to indicate which of the gibberellins we happen to be looking at. Now, as with the oxids, gibberellins can have profound effects on development at very, very low concentrations. So they are plant growth regulators. And as with the gibberellins, I want to just give you a generalized list of the many effects, and this is just, again, the tip of the effects that they produce. The first one is that they stimulate, this is the generalized cell elongation. This is how they were discovered. The second is they can affect flowering. And we're going to talk about that when we talk about flowering. The third is that they have an effect on the state of maturity of plants. And for our discussion, we will characterize these two states as vegetative and as reproductive. That is, there are flowers. And let me give you some examples of gibberellin effects. This is common ivy, which you'll be able to find outside. On a single plant of ivy, you will find these two very different looking shoots. The typical ivy that you know of has these sort of really true ivy leaves. It grows horizontally, procumbent. It cannot flower, but it can make roots. Growing off of this plant is the reproductive portion, where the leaves are very different from these leaves, produces flowers and fruits. It grows upright, so its gravity response has changed, but it cannot root. This particular plant, which is the mature form or the reproductive form, can be caused to revert back to this so-called juvenile form. So we have juvenile and a mature form. This can be caused to revert back to the juvenile form by spraying the plant with gibberellic acid. A profound developmental effect due to this one substance. This is a very widespread phenomenon, juvenility and reproductive state. Here's another example, which is right outside the room. Here's from the eucalyptus grove. This is the typical eucalyptus leaf that you see. It makes flowers. And here is the juvenile form of the eucalyptus. Same tree. Notice how different the leaves are. Again, you can cause this branch to revert to this one by spraying it with gibberellic acid. And finally, what I want to say is that gibberellins have a lot of uses with regard to commercial uses. And the most interesting commercial uses are the following. If you buy grapes in the store, because gibberellin is naturally occurring substances, grapes are frequently sprayed with gibberellins because what this does is it causes the cells to elongate, and when they elongate, they take in more water in the fruits. And so when you find elongated grapes, it's possible that they were grapes that were sprayed with gibberellins. There's no more nutrition, but you pay more because there's more water. For those of you who are gardeners, one of the phenomenon that are controlled by gibberellins is that gardeners like to spray, especially their camellia plants, with gibberellins to produce these large flowers, which are a result of increased cell elongation. This is called gibbing. And finally, the aspect of gibberellins that I want to talk to you about is that gibberellins are used heavily for seed germination. Now, most of you know that if you plant a seed in the ground, it takes a while for that seed to germinate. And that's because most seeds are, in our environment, are conditioned to remain dormant or resistant to whatever conditions are outside until the bad conditions have passed and the seed can germinate and establish itself. The germination of seeds is controlled by gibberellin. So this is controlled by GA. And I just want to point out to you what we know with regard to the features that are involved with germination. Again, we'll take a grass seed that you know of already. At one end of the seed, we have the embryo. The other part that you know of in a monocot grass seed is the endosperm. And then I have to put one additional layer on here for you. And that additional layer is found directly beneath the seed coat surrounding the endosperm. It's called the alurone layer. And this is what happens with regard to the control of seed germination by gibberellins. Number one, a signal, something in the environment, to germinate is received. Signal to germinate. Then the embryo makes and releases GA, which moves to the alurone. Where it stimulates enzymes that break down the endosperm. And the enzyme that most people have studied is one which is known as alpha amylase. This makes the starch available as sugar to the plant, and now we can have germination proceed. Let me show you a couple of examples of GA's effects. Here is a cabbage plant where the internodes, as you know from the lab, are compressed. If you spray GA onto this cabbage plant, you can cause the internodes to elongate, and this is a result of cells being forced to elongate by the GA. Here is the ivy we talked about. Here is the mature form, which can flower. And here is the juvenile form, which can make roots but cannot flower. Now, if we take the mature form and we spray it, we can cause the different different events to occur. We can cause it to go back to the juvenile, or we can cause it to remain an adult. Here is the adult form now, and we've now sprayed it with GA, and we now have caused it to go back to the juvenile form. Finally, I want to show you a couple of the commercial uses which many of you will encounter, especially the guys here in the audience. In this particular case, you can see the differences in the grapes that are elongated, probably an effect of GAs. The GAs are involved in seed germination, and commercially they are important because for the beer making industry, one wants to get the rye seeds, which are monocots, to germinate. You want to get the endosperm, which is stored in the monocot seeds, to convert to sugar, so fermentation can occur. So rye seeds are treated with gibberellic acid, and when they are treated, we have large houses of these seeds, and then this is used to ferment, and this is used as the basis of the brewing industry. So GAs commercially are very important. Okay, we'll see you on Friday, and we'll continue to talk about the gibberellins.